Good afternoon, everybody. Welcome to our UC Ag Expert Talk today. I'm Cheryl Reynolds. I'm with the UC Statewide IPM program. Peter Cosina is also here with us. He'll be running our polls and troubleshooting technical problems if they come up. Please note that this webinar is targeted to growers and agricultural pest management professionals. Master gardeners can certainly benefit from participating, but the pest management methods presented, especially the pesticides, are not to be followed without a clear understanding of their legal use in home environments. Okay, so now I'm going to introduce our speaker. We have Dr. Oleg Daugovish, and he's a strawberry and vegetable crop advisor for the University of California Cooperative Extension, Ventura County. And today he's giving us a review of soil-borne pathogen management in Southern California. So I'm going to pass this over to you.
Okay, well, I'm happy a lot of people could join this afternoon. When Peter asked me to talk about a topic, I was not sure what should I discuss, and instead I decided I'll do a kind of a review of soil-borne pest management, in particular the Southern California and coastal California, and I'm glad to see a lot of folks logging on from Southern California in particular and elsewhere. Well, before I was in Southern California, I sort of traveled all around the place and lived in different places and worked in places like Northern Europe and Florida and Minnesota and Idaho and Nebraska, and every place was different. Every place had some soil-borne pest issues, but California was kind of special that way due to a variety of crops, pests, and growing technologies here. So if you'd go back and work with wheat, well, you could do fallow, you can rotate. If you go to Belgium or Netherlands, so you could do substrate production and avoid those pests and pathogens altogether, growing a clean substrate. But California is different, and over the years, I was fortunate. It's about 20 years. I worked with the plant breeders, plant pathologists, weed scientists, specialists, and advisors, farm advisors all over the state and out of state. And some of them are listed on this first slide, but there are a lot more. And so instead of talking about one particular study or project, I'll try to kind of synthesize and use some of the examples from the research that's been conducted over the years to illustrate some of the key points. I think it would be appropriate to kind of set the table, the stage on the soil-borne issues. I would not talk about nematodes today, and I would not talk about soil-borne insects and the larvae in different stages. And so majority of my discussion there will be about pathogens, a little bit about weeds. But I'd like to set the stage by showing some images. When I came here and talked to the growers and PCAs, soil-borne problems have been here before. They're still here, some of them getting worse. Here's some examples. This is cilantro. We grow quite a bit here in Ventura County, where I'm located. And it dies from this vascular pathogen Fusarium. You could see the roots are all translucent and they're not functioning. The plants will collapse. Our number one vegetable crop here, another big example, is celery. Celery is, in this case, is killed by a Fusarium oxysporum soil fungus, and this particular form of specialis api race 4. Why am I showing race 4? I'll explain in a second, but this is pretty emblematic of what's happening in our production systems. There's a dying celery crop, and taking advantage of the fertilizer and water that's been left behind is our most troublesome weed, yellow nutsedge. Race 4 is somewhat special. It's only been discovered about 6 or 7 years ago. It's very aggressive. It kills the young plants, unlike the other ones. The production of roots is deteriorating as well, and it likes the warm soils. Some of the things that you see on the slide, some of the bullet points, are actually pretty typical to a lot of situations I'm going to talk about today. So some of those things are important to remember. Warm soils oftentimes exacerbate or proliferate disease development. We do have those soils. We grow celery almost year-round, with the exception of celery-free period for about 3 weeks. What's interesting, a lot of those pathogens are specific to host. I think you all remember disease triangle, or you could call it a pyramid. There's a host, which is susceptible, a pathogen, and environmental conditions. Now, to make it a pyramid, you just multiply it by time. If the 3 components of the triangle are there, the pyramid will, with time, develop a disease, create a disease situation. Well, a lot of those pathogens are able to survive and persist in soil for a long time. And that's why they've been here before us and probably be there long after we're gone, because they can actually populate the roots of other crops without causing any damage or any decline of symptomology in those plants. For example, this celery fusarium can grow on strawberry roots, which we do grow a lot of strawberries in Ventura County, and we're going to talk about it quite a bit. The other common theme, with example of celery, which you know that likes wet soil, it's a petiole. Those fields are wet. When you move equipment, that soil gets stuck to your wheels. It gets stuck to everything that's there, your boots. And it doesn't seem like a lot, but you can transport inoculum or a soil pathogen at any level to a different location and keep it spreading. And that's been the case for most of the pathogens we have here in the county and beyond. What's also interesting, at least from pathological perspective, I think the growers and PCAs hate this fact, but it's true. This particular fusarium, even though it's a celery fusarium, it also kills cilantro. It's unfortunate because a lot of our rotation systems will have both. Some of the folks will grow celery and cilantro, and sometimes one after another. I'm mentioning this because cilantro has its own fusarium. It doesn't need extra fusarium to kill it. It has its specific Fusarium axisporum forma specialis coriandri, which is cilantro #1 problem and pathogen. And here's an illustration of this. Why I'm showing you the empty field? Well, it's nearly complete loss. This is interior valley of Ventura County. So yes, it's a warm soil in late spring or summer. And the fusarium can kill cilantro in different stages. But in this case, I think the seed lines, you can see the seed will germinate, and I think they got disease so rapidly that never had a chance to establish. The only thing that's green on this slide, and that's the weeds. Occasionally we have fusarium in spinach. It seems to be the case in heavier soils, which poorly drained. Not as often, maybe. And of course, our #1 crop in the coast, and I think it's true for not just Ventura County, but also Santa Barbara and Monterey and Santa Cruz counties, that strawberry is a key crop, and it's one of the key problem crops for soilborne pathogens because we have a lot of issues. The primary pathogens we encounter that you see on the slider, Fusarium axisporum forma specialis fragaria. So fragaria is strawberries, it's a strawberry fusarium, you can call it in such a manner. And Macrophomina phaseolina, which is another fungus that's causing charcoal rot. They're almost identical if you look at the plants, but they're similar pathogens in the way they cause symptoms. Here's another example. I promised I'll show you a lot of pictures of death and mortality before we talk about management. Here's an interesting field. Susceptible variety, we have that. We have a pathogen, charcoal rot pathogen of Macrophomina phaseolina. Even though it's been fumigated, apparently it wasn't sufficient to kill the pathogen. We have a lot of losses in this field. What was interesting, this actually is a planting during summer, and it's what we call our off-season when we plant in summer to produce fruit during fall and winter. Well, it's a warm soil with pathogen, and we have varieties that's dying. And what to do with this? You have a contract. Grower had a contract to deliver fruit, and interesting situation from this point on, and went out in this field was in September. Use the same beds, and they only pulled the dead plants out to plant new barrow transplants from high elevation nurseries in that same soil, in the same field that you're looking at the slide at, in October, which is typically when we plant our winter berries. That's pretty much all it was needed. Some of it was actually fumigated with methampsodium, but some of them wasn't. And the reality is that none of those plants planted in October had any issues, even though they were susceptible. This exemplifies that soil temperature isn't tremendously important in causing this disease development. And so you could get away with growing those susceptible varieties in very relatively infested soil when the temperatures are 60 degrees or lower. We had about 60 degrees at that time. So it's something to remember as well. So one of the questions, or one of the sort of like frameworks for this is, well, why do soil-borne pathogens persist? Why do we have to deal with them year after year? And then people who inherit the land have to deal with them as well. And our coastal production is partially to blame. We have susceptible hosts planted frequently. That is a polite way to say we have monoculture for some crops like strawberry that go to the same field year after year, sometimes over several decades. If you look at the literature, and I did, for most of those pathogens, the best thing that happens in pathogen life is to have a monoculture of crop that really allows them to persist, multiply efficiently, and do well in those sites. And they persist in soil for several years, even in the absence of hosts, which happens here too. The issue is also for us is lack of affordable and effective control tools. Now, maybe I'll at this point mention that back in the day, we used to have methyl bromide. We used to have, very briefly, methyl iodide, excellent fumigants that made things very easy. They kill all the living organisms in soil. You have a clean slate. It can be populated with a beneficial if you inclined to do so. But basically, it takes care of all these issues, and they were very effective. There's a lot of research been done. Unfortunately, it's all in the history books now. And I'll talk about the tools the rest of the time. But the other problem I see, and I think it's true for some of the minor crops, we don't have cultivars with the resistance. We don't have enough breeding effort going into developing those cultivars that's going to have a commercial quality vegetable or fruit that's has resistance to those pathogens. We're fortunate to have a good breeding program for strawberry, whether it's UC Davis, the public program, or we have very good proprietary varieties that have the same objective of breeding towards disease resistance. But some crops like cilantro or even celery, it's hard to keep up with those diseases. That would be the best solution, have a cultivar that's can be grown in the presence of pathogen. The other issue is it's difficult to invest to a long-term management. You have to be certain that's going to pay off. If you have a short-term lease on the land, are you willing to fumigate? Are you willing to invest into improved drainage? So you have better conditions for crop growth and lesser disease development. Those are big questions. It's a fast turnaround. It's a rapid business, so it is hard to make those long-term plans and be successful. Now, I mentioned, I think at least a couple of times, increasing soil temperatures. Our soils on average warmer than 20 or 30 years ago. It seems that way, and maybe that's the direction we're going. Does it mean we're going to have more disease more frequently? That's an interesting discussion to be had. That's another way to kind of epitomize our challenges in production. We have this very expensive land. This is Ventura, the town I work in and live. We have this nice harbor with multi-million-dollar homes. We have foothills of the mountains, likewise a pretty attractive area. And we're struggling to maintain our agriculture, stuck here in the corner between the two. I like this slide because it shows in a pleasant way the pressures that agriculture experience, whether it's we're trying to use fumigant, or if agriculture makes noise, or is there a runoff or pollution. You got to put pest management in perspective of all the different pressures regulatory pressures, foreign competition, lack of labor, expenses for labor, all those other things that come into play. So I'm talking mostly about strawberry because it's a number one crop. It's a crop of fumigate. It still has about $2 billion value in the state of California, and only about 10% is organic. So we have to deal with both fumigant and non-fumigant technologies to address some of the soil-borne issues that I mentioned in the previous slides. Not to be forgotten, I'd like to also bring our top weed, yellow nutsedge. We do have occasionally Cyperus rotundus, which is purple nutsedge, but majority of our fields have Cyperus esculentus. And actually, it follows a lot of the same patterns. It likes warm soil. It can grow almost year-round, but does best, grows faster, it produces tubers faster, it produces faster when the soils are warm. And we really don't have a lot of good tools. To be exact, we have very few tools for yellow nutsedge management. So as I spend the rest of the time talking about some of the technologies or approaches that we evaluated over the years, just to kind of throw it out there, I like to use the rule of 3 E's. Whatever you're trying to implement or test, it has to be effective, it has to work, it has to be economical, it has to be environmentally acceptable. You can throw anything you'd like to do in these 3 categories and see if they all check out. For example, methyl bromide, it was very effective. It was destroying ozone layer, so it was phased out, and it was also getting very expensive. So during the rest of the time, and I have some bullet points here, I'm going to talk about precision application based on field distribution, and I'll talk about fumigants, but truly, any precision we can gain with our tools is beneficial, whether we're planting a different cultivar, we're applying steam, we're doing fumigation, or whatever decision we're making. If we have precision in how we approach the decision, it will be helpful. I'll talk about ASD, which is anaerobic soil disinfestation. Solarization, I wouldn't talk much about because it can be very effective in the hot climates and in interior parts of the state and elsewhere, but on the coast, where it grows vegetables and berries, it's marginally effective and usually not cost effective. Steam, I'll mention that briefly towards the end of this talk. Biocides, I'll discuss that as well. Cultivar resistance is obviously a key, and I'll have a bit of time on that. Production of substrates, and finally, production when soils are cool. I think that's a theme that's been I've been touching on so far, and I think it's going to be important to mention. So there's different approaches. It's clear we don't have a silver bullet anymore. So we've been looking at all these different things and see if we can combine them, which of them are effective, which were economical, which ones are acceptable environmentally, and which one can be adopted, actually used. So first I'll talk about fumigation. Well, with all the changes that happened in the last 15 or 20 years, we're mostly applying chlorpicrin these days, and MITC generator fumigants such as metam-sodium, metam-potassium. Some people come and say, hey, you shouldn't fumigate. Fumigation is bad, it's expensive, it's toxic, people don't like it, whatever. Well, that's great. This is a slide exemplifying the difference between the bed that was fumigated, the very first one on the slide, and the rest of the field that wasn't fumigated. And it's rare to see a pathogen killing plants as effectively as you see on this slide. In fact, I had plant pathologists in this field, Steve Koike, Tom Gordon, others from UC and elsewhere, and they just couldn't believe it. The strawberry has been killed nearly 100%. In fact, in that field, and you'd wonder, well, if it's a fungus, and this is Fusarium oxyscrum, a fungus, could use a fungicide. And we tried that. We tried pre-plant dip, applying for drip, and none of this stuff worked. But this is a good example, and I have many other examples showing how fumigation is actually very beneficial. And this is a high value investment, so there's no room for error, and definitely not for plant loss. So with fumigation, we predominantly do what is the most affordable way to fumigate, which is through drip. So fumigant is injected in the constructed bed via drip line, gets up for emitters and travels through the bed profile. And what's been a consistent pattern, that efficacy of the fumigant diminishes with the distance from the drip lines that deliver the fumigant. Pathogen survivorship also happens not only away from the drip emitters, but also in any plant material that harbors a pathogen. For example, strawberry crowns that are infested with pathogens are difficult to penetrate, and they kind of serve as a harbor or reservoir for pathogen that prevents it from being in contact with the fumigant, which is effective, but we just can't get to it, at least not at lethal concentration. So that's what we do. It's one of the reasons we still have a lot of those problems with pathogens. As I mentioned, we will talk about it. Flat fumigation, on the other hand, is very effective. That is, the whole field is fumigated with the shank injections and tarped instantly. So the whole volume of soil and depth is fumigated more or less uniformly. Over the years, we looked at pathogens in soil and crowns. It's close to 95% effective. It's not perfect, but it's the best thing we have if you need to control the pathogens and weeds in a flat field. It's expensive. It's $3,500 plus, probably more today already. So it's an expensive but effective tool. This is one slide that exemplifies the issue with drip fumigation. In this particular case, we came to the end of the season, end of strawberry season beds, and used existing drip tapes to inject, this is metamsodium. Right away, you could tell that after about, I think it's 2 or 3 days, the yellow nutsedge that's in those beds occupying, competing with strawberries is getting yellow. But it's getting yellow near the drip lines those are the mother plants that have been grown here. If you look at the center of the bed, I hope you can see it, it's a lot greener, suggesting maybe that's the fumigant coming out from the drip tapes did a pretty decent job on the yellow nutsedge on the bed shoulders where the drip lines are located. There was 2 drip lines per bed, but it wasn't translocated and moved enough to the centers of the bed. Well, so what? I mean, this is a yellow nutsedge, we're going to disc down and forget about it. The problem was all those plants, once they reach 4 or 5 leaf stage, produce tubers that'd be active for 3 years in that soil and cause problems for at least 3 years. And if you look at the tubers, and usually we do this kind of projects, we bury known amount of inoculum, whether it's a pathogen inoculum, a crown, tissue, or in this case, yellow nutsedge tubers. We bury them in different places and see how well they survive. So in this diagram, we have 2 depth, 15 and 30 centimeters, and we have kind of a diagram of a strawberry, but with 4 rows of plants and 2 drip lines marked with circles. So after applying metamsodium, we see that survivorship of tubers from yellow nutsedge is 0 under the drip tape, just kind of like what we saw with the mother plants getting yellow and declining. So that's good news. But we move away from the drip line to the center of the bed and also to greater depth. We're doing an okay job at their survivorship. About 14% of tubers survived in those areas. It sounds like still pretty good control, but anyone of you know who worked with yellow nutsedges, leaving 14% of nutsedge behind viable, it's bad news because of how quickly it can proliferate and reestablish its site. So over the years, we played with bed fumigation. We know it's a common way to do it here. Can we be more effective and put more drip lines per bed? If it's not 2, 3, we use this sometimes 4, even 5. We try to also go not just across the bed, but also go deeper. The problem with the drip tape, you can only bury it to a certain depth. I think we went as deep as maybe 7.5 or 8 inches. It's difficult to bury it deeper, and it's even more difficult to excavate this drip tape at the end of the season. You can't leave it behind in the soil. But the idea was to try to reach some of those difficult areas with extra drip tapes. This is one of the slides from many studies that we did on this topic, showing a combination situation with 4 drip lines on the left. So we look at the soil surface, and those circles are 4 drip lines, as opposed to 2 drip lines. And the color in this chart it's a concentration exposure index. It basically shows you how well fumigant is distributed at lethal concentration. So the more red color, the more effectively we're delivering the fumigant. And in the blue, it means it's vacant from fumigant fumes, or it basically hasn't been translocated there. So this shows you that, well, more lines helped. What didn't help, we have good distribution, and this is VIF. We use, which is virtually impermeable film. We use totally impermeable film these days. They're all great for concentrating fumigant next to the soil surface, under this film, under the plastic tarp. But as we go deep down, neither one of those two scenarios, and we look at the deeper burial tape as well. We never reached 30 centimeters. Those are the areas of roots, or in this case of strawberry, will grow into and get in contact with pathogen that hasn't been killed by this fumigant. So no matter how hard we try, we get improvements, but we still have quite a bit of survivorship. We have limitations with this approach. Another thing we've been looking at recently, we knew that flat fumigation is the best tool we have. It's just too expensive. Can we use it maybe more accurately? This is a project we have area-wide in multiple counties in California and still going on. And this is one of the sites. On the left, you see a field with different colors over. It's about 10-acre block. So different intensity of red color shows the intensity of mortality due to the Fusarium oxysporum fragaria in this field. So the areas that have a lot of red, a lot of dead plants. The areas that are really light has none or very little. So there are different ways to obtain the maps that can be useful. You can have aerial images at the end of the season or in season. You can have mortality counts. You could take soil samples over the grid, which we did as well. And those soil samples can make you, help you create a map. So what to do with this map? Well, in this particular study, we actually split this into 6 plots. 3 of those plots received a standard flat rate of chlorpicrin 350 pounds per acre. And the plots that were justifiably had less pressure, that we thought we could scale back on a fumigant, received variable rate, or what we call precision. It's pretty coarse precision. You can only do as good of a job as you, in this case, fumigation rig can accomplish. But it's an easy fix. It's something that you could do today. We worked with Trical on this, and their operator was had no issues. If you come to the place where you need to switch the rate from say 300 to 250, it only takes a few seconds for him to push buttons and recalibrate. Those points where the rates, injection rates in the field can be switched, can be entered with a GPS unit, or it can be even flagged ahead of time. You don't have to do any thinking as an operator. So you just switch the rate and move forward. So we didn't have any issues applying it. It's easy to do. So that was the key. Is we can be too precise, but there will be no use if it's not possible to have a tool to address it. Anyway, that was a situation from the previous summer or fall. And so in all plots, whether we had precision fumigation with reduced rates or standard, we have this really nice clean field. And we know flat fumigation does a good job. So all the plots on the right in 2018 compared to 2017 look almost disease-free. There are occasionally dark spots here and there, but we didn't lose any yield. We didn't lose any production by using precision fumigation. And we've been really conservative. We reduced fumigant use about 16% only. And I know we could have done much better than this. But even with 16% reduction, using this map and using variable rate fumigation was cost effective. According to economists that worked with us on this project, analyzed what goes in and what goes out. And they say, well, it was worthwhile. And this is the kind of results we see at multiple locations as the project moves forward. The point here, we're using fumigant as an effective tool, but in reality, you can use perhaps steam or pick a cultivar, or maybe use ASD. I'm going to talk about those technologies. So it's approach of creating an inexpensive and workable map and trying to do in-field variability in how you approach your problem, whether it's a pathogen or say population of nutsedge. Cultivar resistance. Well, when we saw this pathogens first time about 15 years ago, even those old cultivars had different susceptibility. Some of them did just fine in the presence of strawberry fusarium like this Ventana. Camarosa was different. So we knew there was something we could build on and develop into component of the breeding program. Give you a couple of slides from our colleagues at Cal Poly Strawberry Center. They actually have a fields of the pathogens uniformly inoculated, and they test not just the cultivars that already exist or been recently developed, but even advanced selections, both from proprietary programs and from public programs. So it's really good to see, and it's valuable to see great variability. And this is a Macrophamina chocolate rot resistance, or actually we look at mortality due to these pathogens. You don't have to see particular ones, but a couple examples from UC Davis breeding, variety Victor, or variety cultivar, cultivar Victor, cultivar Warrior. That's where they land on this continuum from zero being basically almost very low mortality. We got some cultivars in this zone on the left that would be very promising, such as Warrior, perhaps. And a lot of them have pretty high mortality. They're very susceptible. So a lot of work needs to be done yet. This is Verticillium dahlia, which we don't have as much anymore as we used to, but it's a very common worldwide pathogen. Likewise, you can see quite a bit of a difference. Some of them are very susceptible on the right, and there's a lot of very resistant cultivars with almost no or very little mortality. The second question came around, or one of the many questions came around, is, well, we know there's a pathogen. Does it matter how much pathogen we have in soil? Now we're getting those molecular tools that can identify a pathogen, quantify it relatively rapidly. But what do you do with this data? I mean, if your results came with 0 coliform forming units, maybe it's a happy time in your life. If you have 2,000 CFU, maybe what should you do? Do you need to walk away from this field, fumigate, or find some alternative solution? Well, we wanted to see how some of this new cultivars is being developed with the idea to have resistance actually respond to the pathogen concentrations in soil. Let's give you just one of the seasons of data. This is from, I think, a year or two ago. This is a Fusarium axisperum fragaris, or strawberry fusarium. Now, what you see here is 3 cultivars. The new one, Victor, is the blue line on the bottom. That's what we'd like to see, that with increased level of pathogen or concentration of pathogen in the soil, in the root zone, there's really no response. We have no mortality. Opposite to this cultivar that's moderately susceptible, such as Monterey, is marked by this orange line. It has some mortality, but it's not nearly as bad as very susceptible variety as Albion, which is a gray line here. It gets to 100% high levels. What's important besides mortality, which actually dictates the yield overall, is that Victor, which was recently bred for resistance with UC Davis program, at 0, which is no pathogen, just no inoculum at all, and the highest level of pathogen with inoculum level 4 have the same yields. And the yields of other cultivars were actually not severely affected until we reached the very high levels at level 4, remember 0 being none and 4 being the highest. So those are interesting things to consider. And what we wanted to see is what is the relationship? What if you have a number that in your soil that you need to estimate your potential losses? For example, for Monterey, this log model really shows that there's a relationship with a increased pathogen level. It's gradual, but it's a pretty good fit. This is Albion. This is almost a straight line. The more pathogen inoculum you have in the root zone, the faster your plant's going to die. It was somewhat expected. What was important for us to see is for cultivars with resistance, such as Victor, that were newly developed, there was no relationship between mortality and inoculum density. So we know that it doesn't respond to the levels of pathogens in soil, it's good to know. Now, there's a common thing about these cultivars I'd like to point out that, well, the non-pathogenic fungi colonize root cortex. On this slide, it's shown here, kind of on around of the vascular system, and they kind of grow on the periphery of the root on the outside, and they colonize the roots just as well as this pathogenic fusarium. So, but here is Fusarium fragaria. If already susceptible, the fungus enters the xylem vessels, right? These canals here in the middle, and that's bad news for the plant because they're also used to transport water. And if you see that, it's like I like to visualize it as a straw. If you block the straw, the plant is using to pump water to cool off and to use for photosynthesis. Well, you don't have this water, the plant dries out. And no matter how much water you're putting in the soil, irrigating, it makes things only worse. So obstruction of water flow, it's pretty lethal to plants, and that's how you see those plants desiccating in the field, especially when it gets warm. Late in the season, there's a lot of stress on the plant for fruit loads. Plants are transpiring a lot of water, they can't do it, and they dry out and die quickly in those warm soils. The point I'd like to make that it's great to have cultivar resistance. It's pretty much one of the key components of IPM for us, especially in strawberry, but it doesn't mean we solve the problem. On this chart from Peter Henry and his colleagues, that they looked at varieties which are susceptible, such as Albion, Monterey, and with some of the resistant, but colonization of roots was almost the same. So even though the plants that have resistance do not die, maybe don't even show symptoms at all, they're still there, used as a host for this fusarium pathogen, and which will multiply, it will stay on those roots, and you might not even realize how much you have, but it's multiplying and staying on the site, and you just provided them with a host. There's also growth of the fungus on the living tissue. What I show on this slide, that varieties that are resistant, such as San Andreas, they seem to have less growth than the susceptible Albion, but even crop like raspberry would allow the growth of Fusarium oxysporum, which is shouldn't be a host, because we're talking a very specific pathogen to strawberry. It doesn't do anything to raspberry, but it just grows on the roots of raspberry and is able to colonize them and grow them just like it does in the strawberry. And you might never see any symptoms on raspberry, but it's important to keep this in mind as you rotate. And we oftentimes rotate between raspberry or other caneberries to strawberry. So I guess my point is that, yes, it's great to have those cultivars, but don't expect them to control your soil-borne pathogen. The good news, I guess, if you rotate with cilantro, lettuce, broccoli, and wheat, this particular fusarium seems to be not very good when supporting the on those crops. It's not supported by those crops. So, of course, there was a lot of interest in alternatives to fumigants. Fumigants are expensive. They got a lot of environmental pressure. They're difficult to apply. There are more regulations every year. So, yes, there's been a lot happening on that front. ASD, anaerobic disinfestation of soil, is a technology been used in Japan and Netherlands, and it's been used here for about 14, 15 years, and especially among organic growers. The basic idea is to use a labile carbon source. We use mostly rice bran, have moisture in soil, and plastic mulch for about 2 to 4 weeks. So this carbon source and moisture help create anaerobic conditions. So it's like a fermenting process that prevents any oxygen from being left in the system. So you have this anoxic conditions. What happens during this period, the microbiological species that function and work well in those conditions, they change soil environment in multiple ways. Well, you can probably inhibit some of this fungi with anaerobiosis itself, but also there's some biocontrol going on there's a reduction of pH, there's a lack of oxygen, there's interaction between microbiology of soil, microbes of soil that are pathogenic and non-pathogenic. You have also physical changes in soil because you just added a bunch of carbon. And every time you add a bunch of carbon, your system changes. Sometimes it's better infiltration, sometimes you have better salt leaching. Everything you put in soil usually carries some elements. For example, rice bran has nitrogen and phosphorus. A combination of things is difficult to predict, and it happens over time. So as opposed to methyl bromide or other fumigation where you have a fumigant killing all, we have a really complex mixed bag mode of action here with this technology. You could look at it this way. The bottom line, if you have a green, healthy plant, that's maybe all you need. But is it healthy and green because some of the pathogens in the root zone being suppressed due to anaerobiosis? Is it because it's getting more micronutrients, which became available because you reduced your pH, which is an issue for our calcareous soil? Do you have extra nitrogen from, say, rice bran? Did you improve infiltration and leaching? That happens as well. And so it's complex, but the result can be quite favorable. In fact, if you compare ASD and look at its efficacy, if you have minor pathogens, maybe Rhizoctonia, Cylindrocarpon, no major pathogens, what we would call root nibblers, you can have yields almost the same as in fumigated soil. For the key pathogens, you have about 90% reduction of Verticillium dahlia, which is easy to control with this technique. For Macrophamina charcoal rot, we have some reduction, but it seems to be taken very gradually. Fusarium oxysporum that I mentioned, Fusarium of strawberry, well, you can completely fail if the soils are cold during ASD process. And you can actually exacerbate the problem, providing this carbon as a food for the fungus. But if you have warm soils, you can have some reduction, up to 65%. So it's temperature dependent. Just one slide on the subject showing that ASD can be not just effective for vigor and health of strawberry plants, but also it reduces some weeds. We have clear mulch in this. It's pretty rare to see clear mulch in organic system, but we can reduce weed with this technology as well. This is how it's used. That's this case applied to bare soil, incorporated and mixed, beds are made, then plastic is laid out. And one of the key issues, it's also expensive. You have to have a affordable carbon source that works for ASD perfectly well. You don't have to ship it very far away. We looked at, besides the rice bran, blends, grape pomace, and so on. So far, rice bran be the best. It's the yellow bars on this chart. We'll continue looking at this. It's a separate discussion, but it definitely would be more affordable if we can get something locally that's cheap or free. One point I'd like to bring, it's from our colleagues in Spain. You know, they use a thing called biosolarization, which is similar to ASD, but with a solarization component. And the chart on the bottom shows that even the cultivars respond differently to this technology. For example, a cultivar like Fortuna, in this case, in control without biosolarization, has a lot of mortality due to charcoal, but it responded really well. The blue bars is a lot smaller. If you look for a cultivar like Splendor, it's the opposite. Cultivar Splendor preferred untreated soil compared to this biosolarization. So even this technology can be reflected different by variety of cultivars that we have. As an interesting slide from North Carolina, I'd like to share. You know, we hear about soil health all the time. It's a big hot topic, and there's a scale of soil health that you see on the left of this chart. And this is different treatments than Frank Lowe's and his colleagues in North Carolina applied. What's interesting to see that, as expected, the PIC 60 chlorpicrine, this little black triangle on the bottom, had least healthy soil in the beginning, which is expected with a fumigant compared to all other treatments that I'm not going to discuss here. But then by springtime, which is a few months later, it was actually the healthiest. And there was no much differences later. And you know, soil health is sort of proportional to respiration. It's a very good relationship. The living organisms and creatures in the soil, they live and they respire. And so soil health is proportional to respiration. And we like to have healthy soils, but in this particular example, it doesn't seem like strawberry yield, which is on the left, is responding positively to the health of the soil scale that goes from left to right, 3 to 11. In fact, this line that had no relationship between the yield and the health score of the soil. Interesting to know this, so that we don't, people define it different ways, but it's a different way, interesting way to look at it. Application of steam, very briefly. It's, if you sterilize something in your life, if you boil the water, drink, whatever you do to create heat, it can work really well. It's expensive. And you have to use fuel. It's $4,000+ per acre. You have to have transportation. Efficacy about the same as fumigants for the most part. Target temperature, 70% for 20 minutes. It's been evaluated by Steve Fenimore and his colleagues in Salinas, Central Coast. I think it could be valid in some particularly valuable hotspots, maybe in the nurseries. Maybe there's one location in the field, instead of 10 acres, you can do half an acre and prevent the spread of this pathogen or weed from that particular spot, especially if you're organic. There's a discussion about carbon footprint. You're burning quite a bit of fuel to do this. But it's been effective. This is comparing to a little ITC, non-registered fumigant in California. Steam does pretty well for yellow nutsedge, and actually works well with Dominus. This is verticillium, the Soilborne pathogen. Steam does perfectly well for this. You can see on this chart. Actually, I think I'm getting kind of short in time. I briefly go over biofumigation. And well, if you grow a cover crop of mustard species, it produces a lot of beneficial things to your production system. But one of those had a lot of interest is biofumigation potential. And what it is if you have glucosinolates, which you do in most of the mustard plants, in the presence of water and enzyme, which is already on the plant called myrisonase, they break down and release a cocktail of compounds. One of them is pretty famous, isothiocyanates, ITCs. Well, why is it famous? Because it's a component or active ingredient of methyl ITC or VAPAM or KPAM. But there are other sulfur-containing and ITC compounds in this cocktail that may be helpful. Now, to set things clear, if you grow a very good mustard crop, you would not even approach the commercial rates of VAPAM. The levels of ATCs will be a lot lower, so don't expect the mustard crop to do the same job as a fumigant for you. So we're getting a fraction of it, but it was widely believed that even the small concentrations and all this mixture, a cocktail of products, change microbiology of the soil, which ultimately is responsible for benefits. Now, very briefly, you can apply green biomass. It takes time to grow it. Seed products like seed meal or pellets are available. They're expensive. The idea is with a crop, you break cells. Every cell is a container of the glucosinolates. You put water to hydrolyze it and also to trap the volatiles so they wouldn't escape into the air before they can chance to do something for your soil. We did some studies with romaine lettuce and celery, and we see some benefits, as you see on the slides, compared to, say, bare control of bell beans that promotes Sclerotinia minor. We had much healthier romaine, and it seemed to be the trend. There's a change in microbiology that seemed to be more dramatic after the mustards compared, say, to a cereal like triticale or bell bean. I'm not going to skip those slides. Those are the slides from our current mustard. We looked at and analyzed actually all the components, looks like components in the tissue. And for the most part, we were successful in finding what we needed to find. I'm not going to spend time explaining this. So it's a mixed bag, but I think the positive, if you use mustard in that manner, you can expect improvements in yield, which is really important. You might not reduce verticillium, but maybe reduce Phytophthora coctorum, which we looked at as well, or Sclerotinia minor, not dramatically, but you see some kind of improvement. But it is an investment to grow a cover crop. You have to have mulching equipment. Mustards don't grow well, or they grow too well, I'd say, in summer, and bolt, providing us with very little biomass. On nutsedge, I'm going to just mention one thing. Hand weeding nutsedge is probably one of the worst things you can do. It produces tubers, as you see around the drip line, where it occupies the zone where the strawberry is not able to extract water and nutrients from. And our organic folks have been pretty successful using different types of barriers for nutsedge for 100% control. That is, I think what ended up using waxy paper on beds. It's expensive, but it's a lot cheaper than pulling it by hand. And it prevents production of new tubers because the new plants are not able to photosynthesize and stay under it. Production of substrates, very briefly, it's expensive. In Belgium and maybe Netherlands or other places in Europe, the government subsidizes part of your investment cost, whether it's core or infrastructure or so. But for us, we can get yields, good yields, similar, maybe better than fumigated soil, but you have to have this investment in front. It can be organic because your product as a substrate, like a core or peat moss, can be organic and it's weed and pathogen free. You have efficiency in water use, which is a big deal in our drought situation. I think that's might be the last slide. The point I'm trying to make is we don't have excellent tools anymore. We have to find ways to fit them all together and see if they're economical and sustainable, and also put them in perspective of other challenges we have in agriculture, such as labor, water, land prices, foreign competition, regulations, and so on. So it's nice to be as effective and as efficient in pest management, but sometimes we just can't afford it. I'd like to acknowledge some folks that help with this research and extension work.
If I may, I have a question because you were using an example of mustard incorporation in the soil. I have heard that also broccoli works very well, that in reality, if we would do the crop rotation of strawberry and broccoli, that it could significantly help strawberries. Do you have any opinion about that?
Yes, and actually this type of research was done when rotations were evaluated that included broccoli for strawberry pathogens, especially verticillium, based on the work that Christian Subaro did. And Georgi Muramoto was in charge of those rotation studies and his colleagues. And yeah, they show that there's a gradual improvement with verticillium control and strawberry health, but unfortunately, that's just one pathogen that seems to be susceptible to the broccoli ITC incorporation. So first of all, there's the amount of biomass that produced in broccoli. And secondly, there's the economical considerations. It may be possible to rotate with broccoli if it fits in your production scheme, but in Southern California, 80% of our strawberries go back to strawberries because they're not rotated with crops like this. So with the cole crops. So it can work, yeah, if it's a good fit for you. But there are limitations.
Okay, it looks like there is a question in the chat. So what about the microbiological products like mycorrhizas and trichodermas working with biofumigation?
The question about those, I don't have a research with microbiologicals. There are several folks that do that. I'll point to Surendra Dara. He tried, I think, just about any biological product that exists. There's more research going on with Andrea Biscaro in Ventura in our office. He's looking at biostimulants and just about every product. And my experience, what I've seen in the data, a lot of inconsistency. It's difficult to provide them at an economical scale for the large, say, strawberry field or celery field, and to be able to monitor them and be sure that they're doing the right thing for you at the price and the quantity that you're applying to. So if I'd imagine if there was an effective, cost-effective way to do that, and we're not giving up on this. In fact, we might have a project looking at using bacterial products to control charcoal rot. We would probably have it already adopted because that would be great. We just don't see a consistency good enough to be able to move forward and scale it up to the stage where it can actually use it, or there's no economics, so it doesn't work out well, one way or the other.
Here's a question in the Q&A. You mentioned using labile carbon for anaerobic control. biochar is not labile, would that have a benefit or not?
Well, biochar has a different purpose. It's not labile carbon. It has different functions. It's not going to be a fit in ASD. In fact, a lot of things that we tested that maybe have somewhat of charcoal properties. We use, for example, coffee grounds. Well, they can work well if they break down faster, but if they're charred enough and become more of a biochar, they actually do the opposite effect. They sequester water and nitrogen, and the plants become very deficient following this ASD process because it's been tied up into this product. So it has to have a nitrogen to carbon ratio about 1 to 18. Yeah, nitrogen will be 18 to 1 or 20 to 1. That's a good place to start, but there's no substitution for doing trials. And products like rice bran, feed meal, mids, they seem to work well. Things that we tried with have more carbon, like almond shells, and other products don't seem to do the job. I mean, there's on theory, on paper, some products are supposed to work well, but if you start doing tests and trials, that's a sure way to tell what you can gain. How well do they fit in ASD, first of all, and secondly, how well do they benefit the plants afterwards? Because you can create anaerobic conditions, and yet your plants will not do well afterwards due to, for example, sequestration of nitrogen, plant available nitrogen.
There's a question. Is there any endangered species safe fumigants?
Endangered species in soil, and that's a topic that's be kind of interesting to look at. I think that any modification of soil, soil being basically a big whole dark world of organisms, whether it's worms or bacteria or fungi or actinomycetes or something even bigger, larvae of insects, things change. It's really hard to track them other than them being dead or alive, because anything they introduce in soil, whether it's a fumigant or carbon or mustard ITC, mustard biomass or anything like this, will make this change. And in my opinion, it's really difficult to tell exactly in which point of time and how they interact. We sometimes capture the final result, say mortality or proliferation or changes in pH and so on. But it's, first of all, I guess we have to identify what species are endangered in soil and classify them as such, and then we can do the species by technology interaction to evaluate this. But I don't think anybody looked at it yet.
Okay, so I think that is it for today. Thank you so much for presenting.
Thank you for organizing that and inviting me to do that.
We want to thank everybody for attending today, and I think we'll go ahead and close out. Hope everybody has a good rest of your day.
You too.
Take care.